Okay, so for the next concept we should be discussing, we'll get to quickly talk about what is DNA methylation. DNA methylation. So, now for the DNA methylation, just so you know, it's a key epigenetic mechanism. Alright, so it involves what? Adding a methyl group. Of methylation, you're adding methyl, so you're adding methyl to the DNA. Okay, so you could see this 5MC, okay. All right, so it is typically at a 5 carbon, so that's why they call it the 5MC, 5 carbon of the cytosine residue. That's why they call it the 5 methylene carbon, methyl carbon. Yeah, so this chemical tag doesn't change the DNA sequence, but can silence or activate genes. Okay, so it usually influence your development, influence your health, or sometimes if it is done wrongly, all right, disease could come in. So how it works, we said that it's basically an enzyme that makes this happen. So DNA methyltransferases, okay, like the type 1 or type 3A or type 3B, is the enzyme that is able to add methyl group to the DNA. So target site is usually at the 5PG. That's what the 5 carbon of the cytosine. Effect: it can block transcription factor binding, okay, or recruit proteins that compact chromatin. So this can lead to some genes that are supposed to express themselves, they become silenced. Okay, so biological roles is gene regulation, genomic imprinting, X-chromosome inactivation, transposon suppression, all right, then cell differentiation. So these are just the methylation dynamics, all right. De novo methylation, maintenance methylation, all right, then demethylation. Okay, so the methyl group can also be removed. So clinical relevance, okay, if it is not controlled, it can silence genes that are supposed to stop you from having cancer. You understand? So cancer cells are just growing without any gene to stop them. That's cancer. Neurological disorders too can set in. You can age too fast. Okay, all right, so I think that's just it for the whole concept of DNA methylation.